There are three basic types of receptors associated with the cellular membrane. The first type are G protein-coupled receptors. These receptors have a binding site outside the membrane and are made of 7 transmembrane domains which convey the information from the external ligand to the intracellular carboxyl terminal, which acts as the effector site connected to the G protein. This protein is made up of 3 subunits: alpha, beta, and gamma. These receptors operate in a variety of forms depending on the tissue in which they are found and the stimulatory agonist. For example, upon stimulation, the alpha subunit dissociates from the beta-gamma complex and activates a membrane-bound enzyme adenylate cyclase, which in turn converts ATP to cyclic AMP. Cyclic AMP serves then as an intracellular second messenger to carry out the effect dictated by the extracellular signaling or stimulating hormone. Alternatively, in different conditions, the dissociated beta-gamma complex can activate potassium channels to hyperpolarize cells. The second type of cell surface receptors are those described as channel link receptors, also known as ligand-gated ion channels or ionotropic receptors. These receptors form part of a channel that, upon binding by some type of messenger such as a neurotransmitter, allow passage of ions such as sodium, potassium, calcium, and/or chloride. The receptor is made of several proteins, usually 5, and normally they have a binding site for the agonist and sometimes others for co-agonist or antagonist, which help to regulate more precisely the operation of the channel. The third type of cell surface receptors are enzyme link receptors. As the name indicates, these transmembrane receptors, upon activation by an extracellular ligand, activate enzymatic capabilities within the cell. These receptors can be an individual structure which is activated by an agonist, or 2 receptors closely associated, and once both are bound by agonist, they activate the enzymatic capabilities in the intracellular portion of the receptor. Commonly, these receptors are called receptor tyrosine kinase or RTKs because the enzymatic activity is kinase. That is, they facilitate phosphorylation of other molecules which in turn regulate many cellular processes. The insulin receptor is a classic representative of this type of receptors.